Uh, I'm going to give you an introduction to research data management and also talk a little bit about the concept of fair and hopefully explain a little bit about what is fair enough. Now, obviously, we're all working in science and creating data. And I think a key element of that is how we understand data and hopefully showing the value of data, since particularly these days with the emergence of AI and ML, really one of the key assets of our work is data. Just a little bit about Curlew Research. I'm only a member of just a part of a team here. And these are my colleagues. Many of them have worked in biotechs and pharma. And Curlew Research, as it's doing in ASAP, of which we joined in the spring, we're helping customers and biotechs with their research data management. So we have a range of customers that we've built up over the last 10 years. So to data and what we hope to cover today. So yeah, we're going to talk a little bit about research data management. Many of you might be familiar with that as you build grants and are asked for a sort of a data management plan, which is in the UK and elsewhere a key part of obtaining funding. I'm going to introduce, as I said, some of the FAIR concepts and talk about what we consider to be fair enough, and use the ASAP work, which was started before we joined, but is a great example of being fair enough with an aim to sharing data in a broader community. And as I say, in this slide deck, I've included some references which will stay in the deck that you can look at afterwards. And I know with Matt following to talk more about the science, there's a good overlap during the ASAP workflow around what's coming next. So just some thinking about getting us thinking about what does research data management, and as we join as someone thinking about a new project, this cartoon from Errant Science caught my eye a few weeks ago, or a while back, in the sense of when a new project is started and how we think about data. It's really at the beginning of a project that you really need to set the foundations in place for data. If you let your data get out of hand and let it sort of continue to get out of hand, you end up with the situation on the right. And I really must sort of also set your expectations that there isn't such a thing as a data squirrel. There is no magic data fairy that helps manage your data at the end. So what I'm going to try and show you is how we can make the most of the data from an early stage to hopefully bring real value to it, not just in the cycle that you might be involved in, but in that reuse cycle, which is critically more important now with model building and reuse. One way we like to think of data is using this sort of comet analogy. And so on the right-hand side, one can think about when data is first created, and there's a lot of energy and a lot of work goes on. But actually, when you think of a comet, it's got a long tail. And it is really like that, I feel, that data has a long tail of its own, in that beyond its first creation and in its first usage, we hopefully want to get more from that data because there's been a lot of cost and effort put into creating it. And particularly, as I said, these days of the broader use of models and building models over longer temporal data sets. And we certainly found this in AZ when we were building out the data management platforms, that when we were trying to go back to historic data, we had a huge challenge in trying to understand it because it didn't have the appropriate metadata. So it's ever more important to make the most of the data we create now to allow its reuse in the future. So just sort of redrawing the classic research data lifecycle with more sort of attachments around data and the sort of steps that you need to think about data. So when we think about the beginning of an experiment, we're thinking about experiment design, and we might be defining the sort of the proteins or the genes or anything else that we'll be needing to set up. We need to think about that metadata and the sort of questions that we'll be asking of the data. Similarly, as we go on to collect data, so sort of bottom right hand corner, we need to think about the vocabularies that we're using. So, because without some sort of controlled vocabulary, it's very hard to compare data with similar data or unsimilar data later on. And then you get to the point of actually creating the data and understanding where that data is stored. And that's a key element these days, given the volumes and the complexity of data. Having that as part of your data management plan is critical to be able to both make use of it immediately, but also secure it for the longer term. And similarly, as we get to the sort of analysis and reuse life cycle at the top left, we want to be able to share that data in different ways, not necessarily openly, but be able to sort of potentially restrict access depending on whether it's got personally identifiable information or some other privileges that we need to assert on it. So again, we need to be careful about thinking about our data all the way through this workflow, and hopefully that will be part of something that you do. And I'll show in the references, as I say, some good links to how to build a research data plan that that might help you in the future. So I mentioned FAIR was mentioned in the title, and probably for many of you, FAIR is a buzzword that you may have already heard about. And certainly it's been around. It was actually came about in 2016 through a paper from Wilkinson and Co. and it involved, and to their credit, that they came up with some brilliant concepts and to define what FAIR data is, which but there's also many groups now involved in FAIR, both from an academic perspective and including industry within life science. And the FAIR principles are a set of principles that really are simply principles. So the FAIR principles don't tell you how to do it, but they set out these 15 principles around key concepts. So FAIR stands for findable, accessible, interoperable, and reusable. Now, these concepts on the right-hand side are a little bit of a mouthful unless you're working in data day in, day out. So there's a slightly alternative version that we can look at today and maybe make it a little bit more understandable for real use. So let's take the first example here. So findable, as it suggests, it's all about either you or others being able to find data later on. So it isn't just about the setting up the appropriate creation elements of data, but it's, if we look here on what's a good thing about findable, it's making sure that you put that data in some sort of online database or catalog. And obviously on the bad column on the right, you can see some examples of probably things that you shouldn't do, or I'm hoping you're not doing in your work. If we pick the second one, accessible, that really builds on findable, and it's about making sure that then someone else can get at it, but based on their privileges, etc. And it doesn't necessarily, FAIR data doesn't necessarily mean free data or necessarily even open data. Within any pharmaceutical company, there will be huge teams working on FAIR data these days. And it's all about having the appropriate access levels so that the right people can see it and equally the wrong people can't see it. But you must be able to find the license of it and obviously any restrictions that might be needed. Perhaps one of the more tricky bits of FAIR is something called interoperability, and this is where the vocabularies really come in, and also the data formats that are important. So data formats are maybe easier to comprehend in the sense of if you're building using analytical instruments and you take it off in a proprietary format, that's not really FAIR unless you can translate that format into something which is both readable by others but has that longevity. Hands up all of those of you that still own maybe mini discs or other hard disk formats that you haven't even got a reader for, or VHS as well. So we need to think about the data formats critically, but we also need to understand how we name things, and that's where vocabs come in, or ontologies come in. So for instance, if we're naming our proteins, we're clear that we're using a particular an accession number, so we have an ID associated with the protein. And equally, when we're referring to terminology around genes or other aspects, that we're using common ontologies that people will understand, and restrict restrict the things that you can select, such that you make it much easier for people to map between things created in different labs. And that's equally, one of the more, it's both the more complicated part of FAIR, but it's also one of the critical elements. And then finally, we hopefully get to the reusable bit, and you can even add here reproducible as well, in the sense that if you've done all these steps, then hopefully your data is much more reusable and reproducible if someone was to analyze the data or repeat the experiment themselves. You may be asking yourselves, why doesn't FAIR talk about quality? And I think that's a debate that we can have in the question session later. But really, yes, quality comes next after FAIR, and we have to consider that's partly why being fair enough is a critical decision, because you can't just do the FAIR part, you have to consider the quality part as well. So just thinking about building on this sort of concept of being fair enough, just sort of using these the sort of onion diagram here to explain a little bit about how you could think about being fair within your own work. So first of all, it's really about working out your fairness within your organization or your lab, and then thinking more broadly as you move outwards from the top to organization wide, your collaboration network, and global. As you look on the right-hand side here with the arrow, you'll be going through various stages, and this is where the interoperability is really important. So for instance, if you're publishing to a repository, which I'll cover in the context of ASAP in a minute, that you need to think about the sort of identifiers and the vocabularies that you're using to make that publication step really valuable. So you really, in terms of thinking here, it's very good to think locally, but you need to think in the context of how your data is being used. So yeah, just to pick up on that part of how you go about becoming fair, and how you sort of build out this work, because it's very much a journey, something that one of the sort of founders of the FAIR principles, Carol Goebel at Manchester University, has talked a lot about. So one of the recommendations we have is that in thinking up a FAIR approach and linking it to your research data management plans, is that you really need to engage with the local community and interview scientists and workshops, and as it's shown in here with post-its, to really build out the concept of what is good enough for the organization, because not everyone needs to be fully fair, I believe, at this point, because many of us will be starting from perhaps level 1 in our fairness, and it's really realistic that to be able to move gently or slowly through the steps, rather than to try to take huge leaps, because you want to show the value of FAIR as you go. So we very much recommend moving slowly, or moving orderly through those steps, really building on what are you trying to get out of it. And some questions that you might want to ask yourself when you're building out your sort of FAIR planning and deciding what's fair enough for you is some of the questions that are listed here, in the sense that you have to think about not just yourself as perhaps the data creator, but also how others might use your data, either within your lab, within your project or organization, and equally more broadly. So these are very ways to plan your FAIR journey and plan your FAIR data assessment, such that you can sort of prioritize and focus on the things that are really important. And more on the bottom part, as I said, one of the goals of research is to publish it in different ways, whether that be in a publication or a patent or some other exchange with the collaborators. And again, this ability to choose the right vocab, so that you're not building out your own ways of describing, but you're trying to use public descriptors such as vocabularies. And then equally, depending on your research area, and I'll cover this with ASAP in terms of perhaps if you're working in small molecules or proteins, really picking your public repositories is really valuable, because that public repository gives you a lot of fairness in a way for free, because they're acting as the access point for your work, and they give things an identifier, which is one of the critical elements of FAIR. So really be clear about where you might want to publish your data in one of the global repositories, because that really adds value to what you're doing internally. So a little bit of some references here as well, which I'll, as I say, will be in the slide deck. There have been many projects that have been sort of trying to drive FAIR adoption. One of them that is worth noting is one called FAIR Plus, which was an IMI, IHI public-private initiative within Europe over the last few years, working with FPA, so that's sort of European pharmaceutical companies. And it came up with a number of concepts around sort of how to build a FAIR process, looking at maturity. And one of the bigger deliverables was a FAIR cookbook, which is a really good resource for you if you're thinking about sort of beginning the FAIR journey. And just like a programming cookbook or the original Perl cookbook, it lays out a set of sort of recipes that you can use to start the journey of being more fair. So it as I say, it's taken from it's a public resource with the URL there, and you can use their recipes to, at different points. Some are focused on findable, others on interoperable, and it's a really good starting point for sort of, if you have a challenge, to see how you could adopt that FAIR process. But coming back to the sort of the title of being fair enough, and as I said, making this sort of stepwise journey to be fully fair, or to be more fair, let's say, and we would definitely recommend this sort of stepwise approach to fairness rather than making exponential leaps, because really I think there's a lot of change management in being fair, and encouraging your colleagues and your organization to be fair, that trying to do the jump from A to C is a hard ask. And everyone, I think, that have had success from FAIR will be taking these sort of iterative steps based on a real business problem and so on. But let's try and get to some examples from ASAP to make this sort of come alive. So as I mentioned, we only joined ASAP in the spring, and credit to all those that had started this journey right from the beginning, in the sense of what you can do as part of a fair enough journey is think about what outputs you're creating as part of your work. So these are some examples that the ASAP consortium has already been focusing on, laid out here on its sort of on the pipeline view based on sort of viral targets as rows. So with the sense that each of these outputs could be considered a fair output, and each one of those, for many of them, there is already was chosen a public repository that could act as the sort of the place where that data is recorded. And what it needed is a sort of a fair workflow, which I'll show you here. And as I say, many people have been involved in this workflow from the beginning. But if we start from top left in terms of the structural biology part, one of the benefits of the longevity of the PDB is that it's an excellent fair resource, in the sense that it gives a protein an ID and so on. And many of the things that have been built up in ASAP have involved using the Scarab tool that's been hosted in Oxford with the Oxford team. But already by making that choice of using the PDB, it means that in terms of the sort of the publication part to PDB, you then get an identifier and you get metadata. And that's a really good start to drive some of the other activities that are involved here. Equally, in obviously the workflow around protein production, AdGene again is an excellent resource for storing the plasmid information, and likewise that allows you to get an ID from AdGene. So as you can see, you start to build up identifiers for your outputs, and you can link them together such that you can show how the plasmid and the protein are related, and equally how they are related in some of the other workflows shown below here. So by making some clever or clear choices about your sort of public resources, you can be helping the whole linking of data, because that really is a key element of FAIR. Each step along the FAIR journey is good, but the real value is when you link things together. So if we look bottom left in the more classic DMTA, design, make, test cycle, obviously designs are created, those designs are put into a resource called Manifold from Postera, but then ultimately screening and synthesis happens, and we're using CDD to store that, and protocols.io is an excellent resource for storing assay protocols. But through that, we can then enable our publication step to Kemble, which again is one of these excellent public resources that are FAIR enabled. So with addition of IDs for things like cell lines and other things, one again can easily or relatively easily make your data FAIR for others to use and exploit. Equally, there will be other things that come out of ASAP related to models and code that all can become FAIR, and obviously, for more publications and the like, Zenodo is a good way of getting a DOI for your more paper-based. But again, I think in the long run, we'll see more sort of granular approach to publications, where rather than it just being a paper, you might break out the different subsets to make that more mineable by to make it more usable by large language models and others, because without sort of more structure to the data, it makes it hard to make the most of it. But as I say, as part of this journey in ASAP, it's all about making sure that you can really make these identifiers usable and use public ones. So in summary, we would suggest that a big part of the FAIR journey is definitely about the data, but it's all about the processes and people, and we mustn't forget that in this, particularly when you're beginning from an existing process. You really need to include your scientists and your data stewards, hopefully, in that journey. And we very much prefer the sort of invest and reward model rather than the carrot and stick, because I think one has to show the value of FAIR activities, because sometimes it does take extra effort for the scientist in terms of that long-term gain, not just for themselves, but the ability to reuse data in the future. And as I said, I've probably said many a time to colleagues that, the Colosseum in Rome, it wasn't built in a day, but obviously they worked on it every day. And I think that's a key element of a FAIR journey, that you need to do a little bit by little bits to really show the value and be able to show the value back to different stakeholders and the science community. So in summary, as I said, I think one of the bigger learnings from our work, and obviously that's tied to ASAP, is that picking the right repositories, beginning early in the journey, but accepting not everything can be, perhaps is FAIR to start with, is building out that mentality early and slowly. And then, as I say, focusing on usability for the scientists to reward the data stewards if you have them, or hopefully building out that sort of data steward capability, because that's becoming an increasing element of a successful project. And I think it just remains me to thank and acknowledge many of the people. Obviously, not everyone could be mentioned here, but there's many people from ASAP listed, and equally for FAIR Plus and some of my colleagues in Curly Research. And thank you for your attention.